Hey everyone, welcome back. We are in chapter 27. We're starting here on slide 9. We talked about this previously, but we're going to show it again here. We're talking about nephrons and how we start with filtration of the blood in the kidney, and then how we process our filtrate to eventually produce urine. We're going to take a look at the structures of the nephron. Recall that nephrons are tubes. There are a series of tubes within the kidney. There's tons and tons of these very convoluted tubes. I'm tracing one right now in pink. It starts in the cortex region of the kidney, and it dips down in the medulla, and then it comes back up and then goes back down. So again, starts in the cortex, goes down to the medulla, comes back up to the cortex, goes back down. Why does it do that? Short answer is surface area. With more surface area, there's more room for modifying the filtrate. There's also a difference in cortex versus medulla. It has to do with concentration gradients. We'll talk about that in physiology, something called a countercurrent exchange system. As we've mentioned, the first part of, the blood that comes in to get filtered comes from the afferent arteriole. The afferent arteriole then branches into capillaries known as the glomerulus. You can see I'm tracing here this complex branching of capillaries, just like a capillary bed, but it's ball-shaped. That's the glomerulus. And then we exit any blood that will continue, which there needs to be blood that continues. Blood that continues is in the efferent arteriole. So it's not a venule, it's another arteriole. The first part of the nephron, which I'm going to circle in green, this first part of the nephron that encapsulates the glomerulus, this is called the Bowmans capsule. Bowmans capsule. You might see it called glomerular capsule, same thing. The Bowmans capsule is only found in the cortex. It's only found in the renal cortex. It's made of simple squamous cells. Let me erase my tracing here so you can see it. See these cells here? Simple squamous, simple squamous, simple squamous. Thin, flat cells. These thin, flat cells themselves are not the ones doing the filtering. They're catching the filtrate. So whenever you have a lining, like we saw this with the endocardium of the heart, whenever you have a lining like this or blood vessels, you have simple squamous. So simple squamous epithelium for the Bowmans capsule. I'm going to draw filtrate in pink. If there's something in the blood that needs to be filtered out, it comes here from the afferent arteriole, and then it can go through those fenestrated capillaries into the lumen of the Bowmans capsule. This is the beginning of our tube, and it'll exit continuing into the nephron. When we look at the histology of the kidney, we can identify easily, in my opinion, and I hope that this will be one of the easier things for you to identify. You can more easily identify these Bowmans capsules surrounding glomeruli by looking for these rounded clusters, rounded darker clusters. It looks very different from everything else around it. Those are, well, let me erase this and highlight again. The darker part, that's the glomerulus. And then the white part surrounding it, so let me draw my own thing up here. The lining surrounding here, that's a Bowmans capsule. Oops. simple squamous epithelium surrounding the Bowman's capsule. And you see a white sliver surrounding each glomerulus. That white sliver is the lumen. That's the lumen. That's where filtrate is draining into. It's being filtered into. You're getting water, you're getting ions, you're getting wastes. When we look at histology like this, if we see a bunch of these glomeruli surrounded by Bowman's capsules, we know we're in the cortex of the kidney because you only find Bowman's capsules in the cortex of the kidney. Let me switch over to my drawing. We're going to draw out the nephron in a schematic here in the kidney. What I've already drawn here, is, you can see, here's the capsule. Whoops, let me change colors. Here's the capsule, so that's the outside of the kidney. So anything on top is outside the kidney. This region here, that's our renal cortex. So that's where we're going to find our glomerulus. Here, below that dotted line, that's the renal medulla. The renal medulla has sections to it. We've got renal columns shown here on either side. And we've got renal pyramids here. We've got one shown front and center, a renal pyramid. And that renal pyramid is where urine is going to drain out. I've already drawn within the renal column, I've already drawn a branch of the renal artery. So you can see blood coming in. And I've also drawn a branch of the renal vein in this side, just, so it doesn't get bunched up. But you have renal arteries and vein branches going on either side. And to start, remember our renal artery will continue branching, until we get to our afferent arteriole. Our glomerular capillaries. Leading to our efferent arteriole. So surrounding the glomerular capillaries, surrounding the glomerulus, this is where we're going to find our Bowman's capsule. So let's draw our Bowman's capsule. Simple squamous epithelium. Simple squamous epithelium surrounding the glomerulus. There's our Bowman's capsule, first part of the nephron. Filtrate goes from the glomerulus into the Bowman's capsule. We'll continue adding to this. What we can find here in our Bowman's capsule surrounding the glomerulus are special cells. I'm pointing to them in green. Do you see, and they're colored purple on the diagram? on the illustration. These purplish cells with really long grappling extensions, they look like they're just kind of holding on, surrounding the glomerular capillaries. They are called podocytes, literally means foot cells. Pod, podo means foot. And so these little extensions are feet-like extensions. If you ever look at foothills of a mountain, they come out like that. That's where the name comes from, or what it looks like. What these cells do, they add an additional layer of filtration. If you filter something, you generally want to have multiple layers. We want to ensure that we're taking the right things out. Take a look at this transmission electron micrograph up here. What I'm tracing right now are endothelial cells of the blood vessels. So this is simple squamous epithelium. You can see the lumen of our blood vessel where plasma is in here. And you see pores. Those fenestrations here, where stuff can filter out. Our Bowmans capsule lumen is out here. So we want to get filtrate out here, but we have one more layer, and that's what I'm going to color in orange. These little extensions are. These are parts of the podocytes. These things here. So if filtrate is to leave the blood, we need it to go past those pores, then past those foot processes of the podocytes. It needs to get through here like in a Plinko game, if you know what Plinko is. In this illustration over here, top right, again, if something's inside of the capillaries of the glomerulus, it needs to exit past the podocytes into the Bowmans capsule. 2 layers of filtration. There's actually a couple more, but you'll learn that in physio. Podocytes allow for an additional layer of filtration. Take a look at the bottom right. Here is a closer look at those at the Bowmans capsule and glomerulus. Let me color in. This is the glomerulus. All of this. You can't see specifically which ones are blood vessel cells or which ones are podocytes, but sorry, you can't read that, can you? There you go. Here is the glomerulus, and then here are the simple squamous epithelium of Bowmans capsule cells. There's a lot of stuff around it, and that's what we're going to get to next. So we formed our filtrate. Back to that refrigerator analogy, we've removed everything from the refrigerator. Well, not really everything, like red blood cells haven't been removed, but anything that was small enough, that anything that could fit through those pores and through the slits of the podocytes has now exited. One more reason, let me go back. One more reason why we need an additional layer. We don't want to filter so much. We don't want to lose too much water. Otherwise, we'll have no blood left. We're only filtering some at a time. We're constantly filtering all day, every day. As long as you're alive, you're constantly filtering with your kidneys, but you don't want to remove so much that you have no fluid left. And so these podocytes help slow down, but still be picky about what you're filtering. The next portion of the nephron that we enter is called the proximal convoluted tubule. It's a tubule. It's convoluted. It's twisty. and it's proximal to the Bowmans capsule in terms of the pathway. Proximal convoluted tubule. Take a look up here. What I'm coloring in green, that's the Bowmans capsule. What I'm coloring in pink, this is the proximal convoluted tubule. As you can see, it's very twisty. How can we recognize the proximal convoluted tubule in histology? It's not simple squamous like the Bowmans capsule, it's simple cuboidal. And there's microvilli facing the lumen on the apical side. Microvilli means increasing surface area for the purposes, in this case, of absorption. The function of the proximal convoluted tubule is to reabsorb water, reabsorb nutrients. We want to save water. Another major function of the kidney is to conserve water. We are dry land animals. We don't want to dry out. Save as much water as possible. We don't want to urinate any more water than we need to. And we want to absorb those nutrients like glucose. Glucose is super useful. We do not want to get rid of it. So take it back. So we're putting stuff back in the fridge. You can identify, take a look at the histology. All those tubes found in the renal cortex, so surrounding the glomerulus and Bowmans capsule. You can see a tube here. This one happens to be cut long. This one's cut not as long. But look at how much fuzzier it is in here. Look for fuzzy lumens and simple cuboidal epithelium. Fuzzy lumen, simple cuboidal. We'll contrast that with a clearer lumen. Clearer lumens are a different tubule. If we're in the cortex and you see simple cuboidal epithelium and it's fuzzy, that's the proximal convoluted tubule. Let's switch back to our drawing. Let's draw the next part. I'm going to simplify this and not draw it convoluted because that will be a huge pain. It'll get really confusing. But we're still in the cortex and we have simple cuboidal epithelium. So here's my simple cuboidal. These are these are, this is the proximal convoluted tubule. We reabsorb things. Sorry, let me draw filtration. Filtration happened here. That was step one. Filtration. From the proximal convoluted tubule out to these capillaries that you see here, that's reabsorption. Filtration is of wastes of excess water ions. Step two, reabsorption, we're absorbing nutrients and water back to the blood vessels. Since we're reabsorbing, that means we need capillaries again. We're not reabsorbing to the glomerular capillaries, we're reabsorbing to another set of capillaries. These capillaries are called peritubular capillaries. Peritubular capillaries. This is the second set of capillaries in our renal portal system. Glomerulus glomerular capillaries are fenestrated for filtration. Paratubular capillaries, they're continuous. They're the regular kind of capillaries and therefore absorption in this case. All right. After we've gone from the Bowmans capsule to the proximal convoluted tubule, now we go dip down into the medulla. And this dip, because we go down and then back up, this dip is called the loop of Henle. Loop of Henle. We can divide up the loop of Henle into two parts. One part is the part that dips down. This is called the descending limb. It's descending because the filtrate is going down and deeper into the medulla. This part can be distinguished because it has simple squamous epithelium. What's unique about this area is that we are reabsorbing water to blood vessels. These capillaries are known as the vasorecta. I'll point this out. Oopsies. These are capillaries. These blood vessels here are known as the vasorecta. Water is reabsorbed in the descending limb. Where's our descending limb? Okay, there we go. In the next part, which I'm going to color in pink, it gets thicker. This next part is called the ascending limb. This is simple cuboidal. No microvilli, just simple cuboidal. Here, ions are being reabsorbed. Things like sodium, potassium, these ions are being reabsorbed. Actually, potassium might not be the best example. Let's just go with sodium for now. Sodium can be reabsorbed in other ions. This is, once again, only found in the medulla. It is not in the cortex. Proximal convoluted tubule, Bowmans capsule, those are the cortex. Loop of Henle are in the medulla. And within the medulla, you can see the difference. Simple cuboidal for the ascending limb, more squamous shape for the descending limb. Let's go to our diagram, our drawing, and let's draw this. So we're dipping now into the medulla. This is a very simplified drawing. I haven't drawn anything convoluted. So here we are going down. And then coming back up. It's simple squamous on the way down. And it's more cuboidal on the way back up. descending limb of a loop of Henle, and ascending limb of a loop of Henle. Now we've made it. Bowman's capsule, proximal convoluted tubule, descending limb, ascending limb. Descending limb, we've only absorbed water. Ascending limb, we've absorbed ions. Oh, and I forgot to point out one more thing. So I'm going to go back to my drawing. I forgot to point out the vasorecta. So let's point out the vasorecta. I've already drawn it. I just need to point it out. Vasorecta are capillaries. They're an extension of the peritubular capillaries, but they dip down. You can see it dips down into the medulla. So we get reabsorption of water and ions at these two different points. All right, so now we're getting to the next part, and we've come back up to the cortex. We've come back up to the cortex. It's the distal convoluted tubule. It's distal, not in distance, like physical distance from the Bowman's capsule. It's distal in terms of the pathway. You can see that it's actually very close to the Bowman's capsule. We've come right back up next to where the Bowman's capsule is, but it's just farther in the path, distal convoluted tubule. It's smaller, generally stains lighter, and there's no microvilli, so it's less fuzzy. You can see, here's a great example. You can see how clear the white space is here. It's less fuzzy, while the proximal convoluted tubule has a lot of fuzziness in the inside. What are we doing here in the distal convoluted tubule? We're still absorbing things. We're still absorbing ions and water, but now we have an additional function. A special thing happens here. We are secreting ions, acids, drugs, and toxins. We can actively remove things from the blood, from the peritubular capillaries, into the nephron. So here's a second location of where things are added from blood to the nephron lumen. Say that again. The first location where we've added things from the blood to the nephron lumen, that was at the Bowman's capsule. The second location to add things from blood to lumen is here in the distal convoluted tubule. This is targeted secretion. Meaning it's unlike filtration. Filtration is anything that could fit through the hole would leave. Here, we're using special protein transporters to remove very specific things from the blood into the nephron lumen. The way I'm going to draw it, let's actually draw it right now, and then I'll come back. So in a very simplified way, this is not anatomically accurate. I'm drawing my distal convoluted tubule, simple cuboidal epithelium. I had to draw microvilli for the proximal. We are getting some absorption. We are getting some absorption, but we're also getting secretion. So we're adding things to the filtrate. That's why we don't call it urine yet, because we're not done doing things to this filtrate. Now, the way I've drawn this is very simplified. I'm going to trace the path once more. Filtrate comes in this way. We go down, come up, come this way. That's how it looks in this very simplified drawing. What actually happens, because these tubes are so convoluted, is that we come down this way, and the ascending limb is actually over here, and the distal convoluted tubule comes right by the Bowmans capsule. This is the actual location of the distal convoluted tubule. That's the actual location. It's right in between the afferent and efferent arterioles, next to the Bowmans capsule. There is a term for this region. The fact that the distal convoluted tubule is right next to everything, right next to these afferent and efferent arterioles, this is called the juxta means next to, juxtaglomerular complex. Juxtaglomerular complex. Take a look over here. Bowmans capsule, proximal convoluted tubule, descending limb, ascending limb, and look at how the distal convoluted tubule comes right next to the afferent and efferent arterioles. The distal convoluted tubule passes right by the Bowmans capsule. It does this because there are special cells here that sense filtration rate. You'll learn more about that in physio. But there are special cells here in the distal convoluted tubule that sense how much we're filtering at a given time. We want to regulate filtration rate. It's really important that we are constantly filtering no matter what our blood pressure is. Blood pressure influences your filtration rate. We don't want to filter too much. We don't want to filter too little. So depending on what our blood pressure is, we need to change how much we're filtering. We can change the size of cells surrounding the glomerulus. We can influence the smooth muscle in the arterioles to change how much blood is coming in, which influences filtration rate. There are special cells here that we're not going to discuss. So this juxtaglomerular complex secretes hormones to regulate blood pressure and influence filtration rate. Still more happens to our filtrate. We're not done. Bowmans capsule, proximal convoluted tubule, descending limb, ascending limb, distal convoluted tubule. And now we're at this portion right here. This tiny portion is called a connecting tubule. A connecting tubule connects many nephrons. You can see one here, there's another right here, there's another right here, there's another right here. So that means there's a nephron going up somewhere this way, there's a nephron over here, there's a nephron over here. There's many other nephrons, we're just focusing on one. All these connecting tubules lead to a collecting duct. This is a collecting duct. Connecting tubules connect many nephrons into a single collecting duct. Now, there's many collecting ducts, but there's many nephrons to one collecting duct. There's convergence. Collecting ducts are really important. This is our last chance to influence filtrate. This is our last chance to take water up. This is our last chance to save any other nutrients we need, like sodium, which can be very useful. If you recall, we've mentioned before special hormones like the antidiuretic hormone, which comes from the posterior pituitary. For water reabsorption, antidiuretic hormone targets the collecting duct, targets cells of the collecting duct, and tells it to reabsorb more water. So if you have a blood vessel out here, the vasorecta specifically, we can take water, it goes through the cell to our blood vessel. Or we can absorb sodium, but to do that, we need a different hormone. That's aldosterone. Remember aldosterone? That comes from zona glomerulosa of the adrenal gland. Aldosterone is for sodium reabsorption. So we're reabsorbing from the collecting duct into the vasorecta. You can see collecting ducts in the medulla in this histology this way. They're longer tubules. They look longer. Let's draw this in our diagram. So I'm drawing one connecting tubule here. There could be another connecting tubule over here, or over here, or over here. There's other nephrons. But then there's one collecting duct that these many nephrons get into. Oops. Oh, no, that's right. This is our connecting tubule. And here is our collecting duct. Collecting ducts are also simple cuboidal. And notice how collecting ducts go back into the medulla. Last chance to absorb anything from the collecting duct to the vasorecta. I know this isn't the most accurate here in terms of drawing, but we'll go there. Last time we can reabsorb stuff. From here, the last tube, we're going to transition to simple columnar. We're leading to the renal papilla, which is off the page. This last part is called the papillary duct. So to recap, we've formed filtrate. We formed filtrate up here in from the glomerulus to the Bowmans capsule. Proximal convoluted tubule, where we've reabsorbed some water and nutrients. Descending limb, where we reabsorb water to the vasorecta. Ascending limb, where we reabsorb ions to the vasorecta. Distal convoluted tubule, where we've reabsorbed water and nutrients and also secreted toxins, drugs, any other targeted wastes. Connecting tubule, we're connecting to the collecting duct. Collecting duct leads to the papillary duct. And the collecting duct, we've reabsorbed more water if needed, or if not, we don't, and if we need to reabsorb more sodium. And that's how we form urine. Let's switch back to the slides. You can see all this here in this mouse kidney. So this is not the same as a human. We only see one renal papilla here. You only see one renal papilla. There'd be several in a human. But what's nice about this is that we can see it all in one picture with histology. Here is our renal cortex. Deeper is our renal medulla. You can see collecting ducts. And within the papilla, we have papillary ducts. And papillary ducts, again, are simple columnar. We drain the urine to the minor calyx, then to the major calyx, renal pelvis, ureter, urinary bladder, out the urethra. A little bit more about the urethra, since I've only barely mentioned it. Urethra, all people have it. It's in the female anatomy. It's shorter because there's no penis. Penis length would increase the length of the urethra. For that reason, it is more common for females to get urinary tract infections. It's more shorter distance, so it's easier for bacteria to travel up that urethra and cause problems. Regardless of the very distal portion of all urethras is non-keratinized stratified squamous epithelium. And it's lined with some muscle to help push the urine out. I've summarized everything here in this diagram. You can see blood coming from the aorta, renal arteries, afferent arterioles, glomerular capillaries, afferent arterioles, peritubular capillaries, and vasorecta, renal veins, inferior vena cava to the right atrium. As we go through, we get filtration. That filtrate moves the proximal convoluted tubule where we get some reabsorption. The filtrate continues in the loop of Henle, where some more stuff can be reabsorbed. In the distal convoluted tubule, we can get more reabsorption. but we can also get secretion. Connecting tubule, collecting duct, papillary duct, and then out of the nephron to the minor calyx, major calyx, renal pelvis, ureters, urinary bladder, urethra. And you excrete, you urinate. Micturition is the technical term for urination. All right, we've gone on a long journey here, but hopefully that refrigerator analogy makes some sense, and we can see how different epithelium types throughout these nephrons help with the process of filtration, reabsorption, secretion, and eventual excretion. Thanks for watching. That's chapter 27, the urinary system. Please let me know if you have any questions, comments, or in the discussion board. I will talk to you all later. Have a good day.